right, thank you. We'll move on to the next presentation. Arnaud, who's my partner here as a host, will be presenting research on cortical sulcal patterns as a window into fetal life.
Okay, thank you very much, Moya. So we all know that a critical mechanism for development learning is neuroplasticity. We have seen several examples today on this important mechanism to improve cognition and intelligence. You all know, I think, this famous study by Dragonski on learning how to jungling with a change of gray matter volume in visual area, with a change after training and some lasting effects after several months. This neuroplasticity can be also observed not only in motor domain, but also in cognitive domain. For instance, in reading, you have some change not on gray matter volume, but also in white matter microstructure, as you can see on this study by De Schotten and colleagues, who reported some change on the FA on several bundles in ex-illiterate and literate. And the comparison between literate and ex-illiterate show that age when learning is done has an important effect. So we see on these two examples that brain can change during development, and there is a critical mechanism that is neuroplasticity. So we know that learning and cognitive activities can shape the brain. And I want to address today another topic, which is, can the brain shape learning and cognition? That is it possible that the brain structure before birth, so during fetal life, can have some lasting long-term effects on cognition and learning? And so a way to assess these issues is either to follow the brain development, as Christine presented, or we can adopt another approach, which is to look at brain features which are stable during development, namely some features that are determined in utero and then don't change during development. And the cortical folding patterns, the way that the cortical folds are spatially arranged, are a good candidate, because these features of the cortex anatomy are determined during in utero life. You can see on the left panel how the brain starts initially as a smooth surface area that becomes more and more folded during the development. And the timing of apparition of the different sulci have a precise timetable, which means that we can use this sulci as a sort of carbon 14 of the development, of the fetal development, which means we will see after, but according to the sulci which is involved, we can have an idea on when it likely happen during in utero life. An important question is to be sure that the cortical folding pattern are stable during development, and so to investigate this issue, we study in collaboration with Jean-François Mangin and Armin Raznau from NIMH some data of longitudinally acquired MRI of healthy participants from around 5 years old to 20 years old. So you can see on the right the distribution of age of the different participants. And for each participant, we looked at the sulcal pattern, and we checked whether these patterns were stable or not during development. So for instance, on the first line, you can see the 3D reconstruction of the participants at different age, at 10, 13, 14, and 16 years old. And we focused, for instance, we can focus on the anterior cingulate region, which on this participant had just a single, you can see with my arrow here, you just have a single cingulate sulcus. And this single cingulate sulcus is present at all age, and there is no change in the pattern. In this participant, in which we had MRI at 11, 17, 19, and 22 years old, you can see another kind of sulcal pattern for this region, which is a cingulate sulcus and an additional paracingulate sulcus, which is a secondary sulcus that can be present or not in humans. And you can see that in this participant, you have a paracingulate sulcus at 11, 17, 19, and 20-year-olds. And what we found is that without any exception, the ACC and also another region we studied after, which is the inferior frontal gyrus, the sulcal pattern did not change between the baseline and the follow-up acquisition, suggesting that this cortical folding pattern is stable during a period during which the anterior cingulate region undergoes a dramatic and very important change in anatomy. So during this period, the brain is very plastic, and even if the brain is plastic, the cortical folding pattern doesn't change. And so using this information, we can now study and look at early neurodevelopmental constraints on inhibitory control in just looking on participants with different sulcal pattern and trying to find the links between difference in cognition. So we first studied healthy preschooler at 5 years old. So we looked at the anterior cingulate cortex with the two different patterns, and we evaluated the inhibitory control using a standard Stroop task, which was adapted to a preschooler. And so, as in Stroop task, there is two conditions: a non-conflict condition in which you have to name the animal's body as quickly as you can. So here it's easy, it's a duck. And in the conflict condition, the head of the animal does not match the body of the animal. And so the participant has to inhibit the automatized recognition of the animal based on the head to activate the recognition based on the body. And if you compute the different time required to perform the two tasks, you have a sort of Stroop score which evaluates the inhibitory control efficiency. And the lower the Stroop score, the higher the inhibitory control. And when we compare the sulcal pattern of the ACC and the Stroop score, we found that participants with asymmetric ACC, namely participants with different ACC pattern in each hemisphere, a single type in an hemisphere and a double parallel type in the other hemisphere, had a lower Stroop score, which means a better inhibitory control than kids with symmetric ACC. We followed these children during 5 years using a longitudinal follow-up, and we measure their inhibitory control using a standard color word Stroop task at 9, and we observe exactly the same pattern, namely children with asymmetric ACC had better inhibitory control than children with symmetric ACC, and because difference in ACC sulcal pattern can be related to difference in fetal life, this finding suggests that fetal life, and in particular fetal life which have an impact on anterior cingulate cortex, have lasting effects on cognition and here on inhibitory control. We also looked at this effect on adults, and we replicated this finding in adults and also in the other region of the inhibitory control network, the inferior frontal gyrus. And we found that around 14% of the variance was explained by the anatomy sulcal pattern of the ACC, and only 3% of the variance was explained by the sulcal pattern of the inferior frontal gyrus. So 3% it's not a lot, but similar to what we can see in genetic with common variant small effect, it seems that frequent variants can have small effects, but they can collectively account for a large portion of variants. If we combine ACC and IFC, you have around 17% of the variants, which is explained by difference in the sulcal pattern in this study. An important question is whether environmental background can or not modulate this early neurodevelopmental constraints. And so we study bilinguals because we know that participants, people who switch from one language to another one, have a sort of natural training of their inhibitory control because you have to inhibit L1 to activate L2. And so our hypothesis was that bilinguals should have different effect of ACC on their inhibitory control. And it's exactly what we've observed. So in monolingual, we observed that participants with asymmetric ACC had lower, had better inhibitory control than monolingual with symmetric ACC. But in bilingual, we observe a different relationship, namely an inverse relationship. Bilingual with symmetric ACC had better inhibitory control than bilingual with asymmetry. So at first view, we thought that there might be a false positive because we expect no difference between symmetry and asymmetry in bilinguals, and we observed in this data some opposite effects. Very recently, there was a paper by Vaughan and colleagues who observed a similar opposite correlation between brain anatomy, here it was parietal gray matter volume, and cognitive control assessed with Simon test in bilingual and monolingual. So it seems that in bilinguals, there's some things that may happen which induce some difference between the brain anatomy and the cognition. And because we were looking at the sulcal pattern, it seems that early brain development can be modulated by bilingualism. We also study another domain of cognition, reading. So the reading, requires a very distributed network on all over the brain, but there is a critical region, which is the visual word form area, the VWFA, which is important input in this network. It's a region which activates only for written words. And this region, the visual word form area, encompass the occipital temporal sulcus that can have two different patterns, either a continuous pattern or this sulcus can be interrupted, as you can see on the left. And what we found is that children at 9 years old who have interrupted sulcus have better reading ability in terms of the number of words correctly read in comparison to children who have a continuous sulcus. So what is the mechanism that can explain this association? We'll see just after. Just to say that we replicated these findings in adults, and we found that adults with interrupted OTS have better reading ability than adults with a continuous sulci, and this relationship holds only for the left hemisphere. We then look at the sulcal interruption. So in participants with interrupted sulci, we found a linear relationships between the length of the interruption and the reading ability. And so our interpretation is that sulcal interruption length is an indirect marker of neuroplastic process because it can be related to the amount of cortical tissue and or the white matter bundles, which means that if you have an interrupted sulcus, perhaps you have more room for plasticity. To finish, I would like to show you some very recent data in which we investigated not only the reading ability, but the receptivity to reading learning. And so we studied preschool children who couldn't read yet, and we followed them during their first year of reading learning. It's a work in collaboration with Steve Masson at Montréal. And so we first check whether the OTS sulcation did not change before and after reading learning. And so, as you can see on the figure, there is no change. Sulcus, which is interrupted, is still interrupted after reading. And we compare the effect of OTS sulcation on reading learning. And we compare two different reading interventions, syllabic or global. And what we observe on the right part of the figure is that if you have an interrupted sulci, which has been found before to be associated to better reading ability, there is no difference between global or syllabic intervention. However, if you have a continuous sulci, you can see an important difference and an important effect of the reading intervention. Global intervention will lead to lower reading learning in comparison to participants who were exposed to syllabic intervention. Thank you.